Hello students, in this session we'll discuss the activation of adaptive immunity or acquired immunity. So you already know that there are two types of cells which are involved in active immunity. One is lymphocyte, second one is antigen presenting cell. So there are two types of lymphocytes, B lymphocytes and T lymphocytes. So what is the role of antigen presenting cells? Every antigen is processed by antigen presenting cells. And you already know that there are three different types of antigen presenting cells. One is macrophages, the second one is B lymphocytes. The processed antigen is presented on the surface of these cells. One is macrophages, another one is B lymphocytes. The T helper cells especially interact with activated presented antigens and become activated. T helper cells especially interact with the presented antigens and become activated, and the activated T helper cells activates B cells and C type cytotoxic cells in a specific manner. The activated B and T cells proliferate and produce clones, and out of these two cells the B lymphocytes produces protective protein molecules, and those protective protein molecules are called antibodies. So the basic features of acquired immunity: they recognize the antigens. Okay. So these antigens are processed by antigen presenting cells, and automatically the antigen soon after the process they activate T helper cells, and T helper cells get sensitized and release a chemical substance called lymphokines, and these lymphokines activates immune system and stimulates B cells to produce a protective protein molecules called antibodies. And you remember, antibody is a glycoprotein produced by plasma cells formed by B lymphocytes. And typical antibodies Y-shaped structure. And these antibodies are produced against the entry of antigens. When you look at the structure of antibody, before we'll discuss the antibodies are class of proteins which are commonly called immunoglobulins, which are produced in response to antigenic stimulation. The antigen produced by B lymphocytes and plasma cells. In fact, B lymphocyte gives rise to plasma cells, and mature plasma cells produce antibody at the rate of 2000 molecules per second. And each antibody is made up of 4 polypeptide chains. And each polypeptide chain is made up of amino acids. So amino acids, there are two regions in each polypeptide chain. One is a variable region, another one is a constant region. And out of 4 polypeptide chain, 2 are heavy chains and 2 are light chains. So more amino acids with more molecular weight. less amino acids with less molecular weight. That means heavy chains have higher molecular weight, whereas light chains have less molecular weight. So polypeptide chains, all the four polypeptide chains are held together with the help of a disulfide bond. Disulfide bond. So the bond to form a Y-shaped structure, okay, Y-shaped structure, and that we called as the antibody. The region holding the arms and stems of antibody we called as a hinge region. Hinge region. Each chain has two distinct regions. One is variable region, another one is constant region. So variable region, here the amino acid sequence varies, whereas in the constant region the amino acid sequence remains constant. The variable region constitutes antigen binding site, that is called paratope, and antibody has antigen binding site. So antibody has antigen binding site, that is called paratope, and antigen possesses antibody binding site, and that is called epitope. And both paratope and epitope both are complementary to each other. So most of the antibodies carries two antigen binding sites. That's why it is called bivalent. Next one. So when you look at the structure, the polypeptide chains are held together with the help of disulfide linkage, and it is made up of light chains and heavy chains. Each chain has two regions, variable region and constant region, and variable region possesses antigen binding site, and that binding site we called as a paratope. Next, we'll talk about antibody. So antibodies, free antibodies perform three very important functions. One is agglutination, second one is opsonization, and third one is neutralization. So agglutination means the clumping of particulate matter including bacteria, viruses. Opsonization means the coating of bacteria to facilitate their subsequent phagocytosis by the cells. Neutralization means they neutralize the toxin released by the bacteria. For example, tetanus toxin. And there are 5 types of antibodies: IgA, IgD, IgE, IgG, IgM, and Ig. Okay, so these are the 5 types of antibodies. So when you talk about these 5 types of antibodies, the major amount of antibodies contributed by IgG. So it stimulates phagocytes and complement system, provides natural passive immunity in the fetus and newborn, and third one, it protects the body fluids. So apart from this, the antibodies, the B lymphocytes are responsible for humoral mediated immunity. Whereas T lymphocytes are responsible for cell mediated immunity. That we'll discuss in the next session. Thank you.